going to talk about a proposal. It's really just a proposal. So this means I'll be grateful if I get some feedback from you. And you can ask questions at any point if you like. So I'm talking about BERT in the context of DNA BERT, which is sort of a subcategory and a potential link to quantum. Okay, so 3 very complex areas, and I'm trying to simplify it as much as possible. But as Einstein said, keep things easy, but not too easy, so not too simple. Yeah, I'll just remind you of the original BERT paper. This is a Google paper from 2018. Attention is all you need. And that's the basic principle behind it. And I'm trying to talk you through it now, how it works. So basically, BERT has 12 encoders. These encoders are actually quite complex, but the core principle is that we are trying to establish relationship between words. In BERT, the words are tokenized, so they're little snippets of words, and not necessarily proper words, but this is just to sort of highlight it. So we have got a sentence here: the dog did not bark, it ate the food silently, and you can see if you link the different words up, you can establish a relationship. And these are weights, actually, it's the strengths between them. So you can see that it relates to the dog, so it has a really big strength. That would be a very strong weight in the network, and silently is not particularly dog-like, so it has a rather weak connection. So here we go in very simple terms. So BERT has been fed a huge amount of data. I think it has been Wikipedia and other sort of publicly available sort of collections of words and Twitter and all of this. And basically, there's a way for the system to self-learn. It's unsupervised learning. The way it's being done is that some of the words are simply covered up. So, and then the model has to guess what might be the word where there is a mask. And that's sort of a random process. And 15% of all words are masked, and then the prediction is trained on this. Obviously, like you all have seen machine learning models, there will be a loss function, and it's kind of via backpropagation. It goes back through the network. You have those 12 encoders basically to get a really good representation. And this is the pre-training mode. And once you have a pre-trained model, you can then actually do more interesting stuff with the model. You can get it to sort of predict the next word. You all have your mobile phone, and when you type in a few letters, it will give us some suggestions, usually not the ones you want, but you come up with a sort of prediction. You can also use it for sentiment classification. You can look at the text and try to work out what has been the mood of the person talking. You can use it for a chatbot, and you can even use it if you have a longer text and you want to create an abstract. You can use BERT for it. So BERT is quite handy. It's part of what we call natural language processing models. Okay, now let's move on to DNA BERT. So the funny thing is that DNA actually isn't all that unlike human language, which you might find strange, but you have little words as well. These words make up proteins or actual instructions, what should be happening. And there are actually, there's a 1994 paper that's showing that DNA is actually, there's a lot of statistical similarities to human language, funny enough. So, okay, I'm just going to talk a bit about DNA now. Okay, so this is sort of what we usually learn in school. Basically, you have the nucleus that's inside the cell. You've got your DNA, and then DNA is transcribed into first pre-messenger RNA, and then bits are cut out, so-called introns, and then you end up with your messenger RNA, which then goes, this is taken outside the nucleus. Here you have ribosomes, and then the messenger RNA is transcribed into, translated into proteins. So just 2 terms you might come across. One is transcription, which basically means the information flows from DNA to RNA. And then translation means something, it's translated in another language, i.e. into amino acids that make peptides or proteins. I'm sure that, yeah, I'm going to come back to this later. That's okay. Right, so, but actually the fact is that most of our DNA is not actually coding for proteins. It's only actually 1 or 2% that makes proteins. The rest is, we don't really quite know what it is. We used to call it junk DNA, which is probably a bit unfair because it's hard to imagine that there's a lot of junk in evolution. So 98 to 99% of our DNA is non-coding, and we only understand a very small part of it. An interesting part, or interesting sort of part of DNA is so-called SNPs. Sorry if I'm bombarding you with lots of terminology. And SNP stands for single nucleotide polymorphism. So these are mutations effectively. And obviously, as most of our DNA is non-coding, 98 to 99%, this is where most of the mutations can be found. And the non-coding DNA has regulatory function. So that means it kind of sits on top of everything else, and like a programming language decides which sort of parts of the DNA can be read and transcribed and translated, sitting there in the background. And I think you have to see it as a network, like a programming language. Okay, and these SNPs are quite important, or these variants, because they are associated with quite a few medical conditions: cancer, autism, heart disease, just a few examples. It's an endless list, and new discoveries are made as we go along. Right, so I'll give you an example of these SNPs. These are meant to represent 4 different people. Each of them has a chromosome from their mother and their father, and you can see that in the place here marked in orange, this is where you find the SNP, and it can be different from person to person. Now, I have to say these SNPs are not necessarily associated with disease, but they can be. So they're quite important from a research perspective. And there are lots of studies where we look at these SNPs and see if we can associate this with any sort of medical condition. Okay. Can you all follow me so far, or any questions, anything you want to ask? Kevin?
Any questions, anybody?
Okay.
Yes, Aidan Challen here. Just the SNPs are in the 98 to 99% non-coding DNA. Is that right?
Yeah, I think they can be everywhere, but they're more likely to be found in the non-coding DNA, simply because most of our DNA is non-coding. That's what I'm trying to say, basically.
Okay.
Okay.
So in this diagram that you're showing, the SNP is the entire character phrase, not just
No.
highlighted?
The SNP is the bit marked in orange. Just
and does that represent just one?
That's just one letter. Yeah, that's why it's called single nucleotide. It's just one letter that is somehow not quite, that's different between these individuals. That's the point, basically. We know there's a difference, but we don't often know if it's actually relevant in terms of a specific medical condition. It may or may not be, and you need lots of statistical tests. You would need people who have the disease and people you don't have, and then look what these these SNPs look like between those individuals. So it's not that straightforward. In the first instance, we just know if you sequence the DNA for these 4 individuals, you find a difference in that spot. Okay, and that's the SNP, the single nucleotide polymorphism.
Thank you.
Okay, you're welcome. Right, sorry, a bit more DNA. So now that you understand a bit of it, you're all experts. So the green bit here, that's what is eventually becoming a protein. Okay, that's the actual gene. And then we have that yellow sequence in front, a so-called cis regulatory element that could be a promoter or transcription factor binding site. Basically, you have to see it as a switch. Okay, so that can activate the gene, and then the gene is basically first transcribed and then translated and makes actual proteins. But nothing is happening without this yellow sequence being activated. Okay, And that's what we're interested in for DNA BERT. Or there would certainly be other bits as well, but the study on DNA BERT focused on those cis elements. Okay, so how do we do the whole thing then for DNA? So very similar to what you've seen before. We're going to use a sort of the human genome. There's a sort of reference genome. You can see here GRCH38P13. Don't ask me what it means, but that's just, they all have very funny names. And it's okay. So we got then this is just a random sequence. I just made this up here. And what happens, like before, you have seen that we create these tokens. We do the same thing here. So this is basically the reading frame. We have A, A, C, G, T, A. What we're doing, we create so-called threemers or k-mers. So 3-letter words, AAC, ACG, CGT, GTA. And this is now fed into the encoder. And again, we are masking up 15%. We are not doing next sentence prediction as we would do for human language. We're just masking it. And then we ask the encoder to learn the sequence that has been covered up. And that's how DNA BERT works in principle. Okay, you can follow me so far? Okay.
Well, I can't say
Okay.
I've taken biochem some years ago. I know it's A to T and C to G. Now it's interesting because obviously this is the progress because we had the human genome project, and I think they went back on it and said that actually now we do think we know, to some certainty what percent DNA. And then there's epigenetics, which is like methylations and all these types of things on the sides of the double helix. And it's also interesting. So when you actually do a DNA test, that only tests for like less than, I think it's either 1% or 0.1% of your actual DNA, because it's only looking for, like Berend has been saying, that we only know certain things about DNA, and we're finding out, is it the case? So basically a lot of the junk is turning into stuff that we do know?
I think a lot of the junk is we don't know, but yeah, it's turning to stuff we start learning to know about. I mean, it's still possible that there might be some sequences which don't have any obvious function, or they might be so redundant, perhaps, even if there's damage. A bit like the human brain, there's some parts, if you damage them and you won't notice this. So there could be some sort of other sequences might compensate, because it's a network. And obviously, like any network, you might have some safeguards to ensure that the whole system doesn't fall apart. So that's possible. And I think that we will learn, because I think network biology is a big thing right now. Okay, so let me just sort of summarize at this point. So non-coding DNA, as I said, is very similar to human language, at least at a statistical level. And then the way we are training the system is by using k-mers. K-mers is a common terminology in genomics, which basically is just how many of these nucleotides you use to create an artificial word, really. And so for the DNA BERT, they used 3 letters to 6 letters, basically. And they have trained 4 models in their paper. And the whole DNA sequence, the whole window is 512 base pairs. And as I said before, 15% of DNA has to be masked for unsupervised learning to take place. And then once you've done this, you have pre-trained your model, you can now start actually looking for a task, an actual prediction. And that could be that you compare a specific disease where you may have a group of people who are perfectly normal. That would be a reference DNA. And then you might have a group of people with a specific condition. And then you could train the DNA BERT basically on making predictions where you first do the training on the top layer. That would be the classifier. You don't have to train the other layers. You have done that. All you need to do is focus on the top layer, which you can just put on top. Okay. Well, we've discussed this. This is just a repetition of what we said before. These are the cis regulatory elements in the main promoters I've been talking about. Right. Now, then there's this paper. I don't know, is some here? amongst us because I
Well,
invited him.
I saw him registering, but I haven't seen him hop on
Yeah,
yet.
you know, basically he published this fantastic paper with some colleagues of his, which were basically they used a quantum layer on top of it. And that basically would be my proposal, because I'm kind of naughty as I am, I'm kind of pinching his idea and bringing all these ideas together, the DNA BERT, but now with the classifier layer would be a quantum layer on top, variational quantum circuit, for example. And it's quite nicely described in his paper. Slightly different because he's sort of trying to deal with security aspects using a network of computers. I wouldn't do this. I just want a quantum layer. And this is basically his quantum circuit. Again, I've taken this from his paper, where basically the way it works, we would have basically 4 qubits, or probably we'll have hopefully some more qubits. This is just an illustration. And then we use angle encoding, where we have rotational gates. And this is followed by so-called U gates, where you can fully parameterize each gate, and that we would sort of randomly initialize it. And then we have C nodes here just to entangle all the gates. And then at the end, we take a measurement. And obviously, then we have to find a loss function as well once we have the measurements. And that's basically the quantum part of it. So it's a hybrid model, like most of the models are. And we would have to think quite hard about how we integrate it, also how many shots we would be running, because as you know, you can't just run because you get a 0 or 1. You have to do thousands of them, and then you average out the zeros and ones to get an 0.5, 0.33, whatever measurement for each of those qubits. Right, so my proposal is therefore to look at some kind of outer layer that would be probably a variational quantum circuit, but there are other quantum gates, and I'm grateful for any suggestions. Obviously, you would have to benchmark this, because maybe it's a pointless exercise, maybe it's not, maybe there's an advantage. Would have to compare it against a classical layer. Other interesting questions, and I looked at some papers, is there a way of compressing the DNA sequence? Big limitation of current quantum computers is obviously the limited number of qubits. At most, you get sort of 200 qubits, and that's only available to a few privileged people. And then the other thing, maybe a quantum input layer. It's worthwhile thinking about it, because if you're projecting into the Hilbert space at an early stage, maybe there's an advantage. And then there are other models. BERT is just one transformer model. There are more specific ones. A recent paper, SNP2VEC, seem to be more specific, because let me go back to the original slide here about these elements. As you can see, you have obviously maternal and paternal chromosomes, and we call this diploid. If you have only one of them, one sequence, that's haploid, and that's what DNA BERT is doing. But the other one, the other transformer, the most recent one, is actually looking at both gene sequences, which makes it a lot more realistic in a clinical setting. So we have to think very hard as well, is BERT the best transformer model? Right, and yeah, so questions.
So obviously this is awesome. It's detailed. So the biggest thing is obviously getting the main points out of it, as far as like, you know, I guess, what is the significance in just in the overall scale, what quantum is doing here in this kind of biochemical DNA application?
Well, the significance is that it might actually potentially improve the accuracy of the predictions, or there could be a speed up. I mean, that's what the research has to be about. That's the purpose, basically, and that's what. Unfortunately, Sam isn't here today to talk about his project, and if he has seen any actual quantum advantage, basically, but there may well be, basically.
Yeah, and for a lot of, and this goes for everybody, for a lot of these scientific papers I've just been researching these past couple of days, is we're just getting the methods down, or basically getting, yeah, methods, analysis, these types of things. So when the good hardware hits, you know, less error, more powerful, then we start to experience, or we start to witness the fruit of the labor of all these types of things that Berend is working on.
Good,
Any
yeah.
questions? Any questions for Berend right now? Go ahead.
Anyone? Any suggestions? I mean, I also take suggestions if anyone has been working with BERT models or transformers.
I
Step
kind
forward.
of chuckle because one of my acquaintances says his name's Bert, and then I tell him every time I see him, I say there's a thing named after you in quantum. Yeah, go ahead, Ron.
So in your this is a prediction that you're looking at, or have you actually tried to put this into practice?
I'm going
As
to
far
put
as
this
using
into practice.
I'm sorry.
I'm going to put it into practice because it's quite doable, because the models are there. It's a pre-trained model. It's not actually impossible to put a quantum layer on top, basically. So what we have to look at really carefully is what we want to train it on, sort of to find a good clinical model and a publicly available library, because you download these libraries for certain gene sequences, and then you can process them.